The webinar will begin in 8 minutes. The webinar will begin in 6 minutes. The webinar will begin in 4 minutes. The webinar will begin in 2 minutes. The webinar will begin in 1 minute. Okay, we're going to get started now. Good day, everyone. My name is Joy Concepcion from QPS, and I will be the moderator for today's webinar. On behalf of QPS's seminar series, I'd like to welcome you to the application of LC-ICP-MS in drug development, presented by Dr. Fred van Hueveln at QPS Netherlands. A few housekeeping comments before we begin. Everyone will be muted during the presentation. Please enter any questions under the control panel. If we cannot get to your question, we will respond to you directly after the webinar. This webinar will be available on demand to all registrants. We will send you an email once the recording is available. And now I would like to introduce our presenter, Dr. Fred van Hueveln, the Director of Elemental Analysis at QPS Netherlands. Fred graduated in inorganic and physical chemistry at the University of Groningen in the Netherlands. For over 15 years, he was involved in materials research at the Energy Center of the Netherlands and obtained his PhD in 1997. In 2004, the ICP-MS was established at QPS, where there is a dedicated group to elemental analysis. In the last 16 years, many ICP-MS studies have been performed with about 40 different elements, from simple total analysis of one specific element to complex speciation studies with simultaneous analysis of free and protein-bound drug compounds. And with that, I would like to welcome Fred.
Okay, thank you, Joy, for your introduction. And also, good afternoon, good morning, everyone. Also from my side, welcome to this bioanalytical online meeting. Thank you all for joining us from different places. In bioanalysis, we simply want to analyze molecules, preferably fast, easy, accurate, and sensitive. However, not all molecules are easy to analyze because of their properties and complexity. In these cases, ICP-MS often can be of a valuable solution. That's why QPS has added ICP-MS to its other analysis methods. So let's first have a look at the topics we want to look at. Of course, we start a little bit with the theory, what is ICP-MS, then why and when should we use it, then experimentally, how does it work in practice, and of course, the most interesting part, what are the different applications that we can use it for, and of course, I will end with the conclusions. ICP-MS, what is it? What is the abbreviation? The word ICP-MS means inductively coupled plasma mass spectrometer. As you can see in the word, there are two parts, the ICP part. Here, the conversion of the molecules into sample ions are treated, and the MS part is the detection of the ions, the so-called mass spectrometer part. If we want to define it in a nutshell, we could have some definitions, which is here given in the box. A molecule in a solid or liquid or gas matrix is transferred into an aerosol, then broke apart into elements in a hot argon plasma at temperatures of 6 to 10,000 degrees C. The resulting elements are then ionized and detected by their different mass charge ratio in the MS. So to remember, in the ICP-MS, we measure only elements. That means that we should always consider an element that is specific enough for our compound. And this element is usually referred to as a tag. In the next slides, we will see at a schematic overview of the method. Here we can see at the left part, here is the sample pumped up from an autosampler to the nebulizer, where the sample is sprayed into the spray chamber, and then the sample is coming into the hot argon plasma, and there it is introduced into the ICP-MS part, where the ions are formed. By using the electrostatic lenses, the ions are attracted to the MS part and are separated here in the quadrupole by the mass charge ratio. What are potential applications and why should we use ICP-MS? We will have a look at that in the next sheets. By definition, we should remember that isotopes are measured in the ICP-MS. Special isotopes or isotope ratio can be used as a fingerprint. I have at the right side given two examples. For instance, the platinum spectrum. Here you can see there are different isotopes, different masses, but also for the gadolinium, which has another fingerprint. At the top, you can see the table, which shows that each of the different elements have several natural isotopes. However, there are also some elements which you can here see, for instance, phosphor, which has no other isotopes than its own mass. From the whole periodic system, there are about 20 elements which have no isotopes other than their own mass. ICP-MS can also be used for as an alternative for mass balance studies instead of expensive radioactive labels. It can also be of a complementary value for other techniques like MSMS, UV, or ELISA, but we can also use it in parallel detection with HPLC UV, LC-MS, or for metabolite profiling. Another potential application is when we use it in combination with immunoassays where the antibody is labeled with a specific element. At the lower side, I have given a figure where you can see that the antibody is labeled by gold. And in this case, the gold will then be detected by the ICP-MS instead of the coloration detection as used as presented in the lower part of the figure. Besides applications as illustrated here, there can also be specific reasons that ICP-MS is the best choice or maybe the only choice. Elements sometimes are present in compounds which are very difficult to ionize. For instance, the platinum compounds like cisplatin, carboplatin, but there are many more. These compounds are very difficult to ionize. However, these compounds consider an element that is very specific. In this example, platinum is present in these compounds. And platinum, as you know, is a heavy metal and can, for that reason, very good analyzed by the ICP-MS. But there can also be other reasons. For instance, if our compound of interest is a Mars or a compound with a very low Mars, then the ICP-MS is the only case where we can measure these kind of compounds. In case you want to have an impression of which kind of metals are used in the pharmaceutical applications, I have given here below a table where you can see that there are a lot of compounds which are used in metallopeptides or metallodrugs and miscellaneous compounds. All these compounds are related to therapeutic areas or to the pharmaceutical areas. So as you can see, there are many applications, metals as well as non-metals. Let's now have a look at the advantages of using ICP-MS in bioanalysis. An advantage is the high sensitivity, a broad dynamic range, more than 6 decades, but also its ability to multiplex. The sensitivity is usually comparable to LC-MS, but for heavy metals, it holds that the heavier the metal, the better the sensitivity. A multi-element analysis, well, in that case, the advantage can be that we can analyze with ICP-MS more than 20 elements within a second. But also isotopic distribution. That means if we look at different isotopes from one element, these can be used as a fingerprint. And that will add more selectivity to our analysis. We can also use it in tissue analysis, biopsies. In that case, we have two possibilities. The tissue can be extracted or we can digest it. In the extraction, the procedure is more or less comparable to LC-MS, but when we can digest the tissue as a whole, then we have a big advantage because we don't have any homogenization issues anymore. The tissue is now homogenized or digested, and only a small part of the digestion liquid can then be used for an analysis of the ICP-MS. Let's now have a look at another advantage. That is the sample processing is very limited for ICP-MS. The so-called dilute and shoot possibility for instance, plasma and serum and urine is a big advantage because we only need to dilute these matrices with an internal standard working solution, and even whole blood can sometimes be directly measured. That means that small sample volumes are possible, even down to 1 microliter. An example is given here at the right side. Here you can see the cross section of the ear, the inner ear, and for a sponsor we have analyzed, for instance, cochlear tissue, as well as the perilymph, which is the inner ear liquid, from mouse study samples. Here the samples were between 1 and 5 milligram. That you can, that is really very small. It's about, you can consider it as a sugar crystal. In these small samples, we were even able to analyze platinum down to LLQ values of 0.2 nanogram per gram tissue or nanogram per mil of the perilymph liquid. Another advantage is the short analysis times, which gives us the possibility of high throughput when we are using 384 well plates. And also very interesting is because of the simple processing and that there is no extraction needed like LC-MS. That means no liquid-liquid or solid phase extraction. That means that recovery is no issue at all. In the next slides, we will focus on some of the experimental aspects of ICP-MS. There are different modes that can be used for ICP-MS. At the top, we see the two possibilities for total analysis. That means for total, we just measure the element in a form that where we cannot make any discrimination between the element from the parent drug or from a metabolite. So that means all the element that is present in the sample is measured, irrespective of where it is coming from. Then there are two possibilities. We can have here an SPS autosampler. This is used in the mode continuous flow analysis. In that case, the sample is continuously sucked up and pumped by a peristaltic pump to the nebulizer, and then it is sent to the ICP-MS. However, we can also use a well plate of a conventional HPLC. In that case, the sample is injected from the well plate and it is injected into a mobile phase, and then it is sent to the HPLC, to the ICP-MS. But just notice that in this case, there is no column used. We just use the autosampler from the HPLC. A third possibility, but completely different from the total analysis, is now LC-ICP-MS. In this case, we just do a separation on a HPLC, comparable to what happens at LC-MS, and then the separation of our parent from, for instance, a metabolite or from other present compounds which have the same element enables us to look specifically at our parent or specifically at a metabolite. So after separation, all the different compounds from each other in the samples are then sent to the ICP-MS. If you look at the resulting the results from these different ICP modes, then at the top for the continuous flow analysis, we have just a number of counts. When we use it as flow injection mode, no column, so that means we have some kind of a peak, but as you can see, there is no retention because there is no column present, so the peak is just at the left side of this chromatogram. It's not a real chromatogram, but just consider it as. And in the LC-MS mode, of course, we have a real chromatogram because we have a separation. And here you can see that there is a retention. But what is more interesting now, we can see this is the peak of our parent, but we see here also a peak at the right side and some two small peaks at the left side. If, for instance, this peak was platinum, then this peak is probably from a metabolite or a degradation product. So this gives much more information, but the question is in which information are we interested, and that will determine which of the three modes is our ICP-MS mode that we will use. If you look at some characteristics at the top, there the procedure is the measurement is slow, about 5 to 7 minutes per sample, low throughput. The output is a number, but we have high sensitivity and minimum carryover because there are no metal elements in this auto sampler, and the minimum sample volume is 0.5 to 0.8 mil. Usually this is the standard sample, that is the standard auto sampler that is delivered with an ICP-MS. However, if we use a normal HPLC as an auto sampler, then we have, because of using the well plate sampling, and the injection is then giving us a 1 minute per sample high throughput, which enables us to have about 500 samples per day. Of course, that is more or less determined by the specific assay, and but this is what is in some situations possible. The output is now a gram, so it is important to realize that we need to integrate this peak to obtain the result. Medium to high sensitivity, carry over only for specific metals, in particular those metals that are present in the HPLC system. In the lower part, where we have now LC-ICP-MS, also referred to as speciation. Here we have a runtime of about 10 to 30 minutes, but it is totally dependent on the application, so it can even be more. But the output is, of course, giving us much more information now, and we have now real chromatogram, medium to high sensitivity, carryover for specific metals, or there is no carryover when we use a bioinert system and UPLC. And very recently we have acquired such a bioinert system at QPS, where we have now the possibility to decrease our endogenous background of specific metals. Also, the minimum sample volume is here 20 microliter. Let's have now a short look at the different machines in our lab. Here you can see the different, the 3 ICP-MS machines. Also here the SPS auto sampler here at the top, and at the bottom you see the conventional HPLC system. All 3 systems are equipped with an SPS auto sampler. We have 2 HPLCs as front end, 1 UPLC bioinert system as a front end, and for the total analysis we have the SPS auto sampler already mentioned, which can be used only in the continuous flow analysis mode. This means that this combination of ICP-MS machines and auto samplers gives us a very high capacity, but also flexibility for the different kind of ICP-MS studies. Now it's time to look at some applications, because that's of course the reason why we have these instruments. One of the most widely used application in drug development is PK for all kind of applications. We can do this as a total element analysis. Already mentioned, in that case we don't see any difference between parent metabolites, but that doesn't matter in these cases if we are sure that there are no metabolites or degradation products. So in that case the total element analysis is certainly the preferred situation because of its high throughput possibility and simplicity. The most used elements I have mentioned here for PK applications that we did in the last 14-15 years are platinum, iron, copper, gadolinium, zircon. Above this we have up to now about measured 40 different elements. The studies that we use are explorative but also GLP compliant methods, and all full validations for GLP applications are all done according to the latest FDA and EMA guidelines. An example of a PK application of a preclinical study here is given in two figures where we have analyzed rat plasma sample at the left and rat tissue samples at the right side. Here we can see the plasma curves for two dosings, but we can also see here for the free analysis as well as the total analysis. The free, in this case, it was a platinum compound. The free platinum was obtained by ultrafiltration of the plasma samples. In this case, that means that we're here looking at the platinum compound, which is effective, because this is reflecting the platinum compound that is bound to be mostly to albumin, which is usually considered as not effective in the body. At the right side, we can see that there is a difference between the different tissues. So this gives us the possibility of looking at the PK profiles at different situations. Another example here is that we can also look at element analysis, but then chromatographic separations of our target element from the parent drug and metabolite. This is comparable to LC-MS, but we have another detector which is now looking at elements instead of molecules. And another difference is that for ICP-MS we usually have a high preference for aqueous mobile phases. There can be an organic phase used, but that's usually that is limited to say 40 or 50%, but depends a little bit on the application. At the figure, we can now see that in this case we can see the oxalic platin, but we can also see that this is separated from the metabolite, the agrodasplatin, and also from the albumin-bound oxalic platin. And even then, we can see that it also has an influence at different times in the PK profile, but also from the ultrafiltrate, the free compound. This was also studied at different times for one situation where we can see that the PK profiles, but also the different parent and metabolite peaks are differing and shifting in time. So this enables us to give much information for metabolite studies. And of course, this possibility, the LC-ICP-MS can also use and is used by our lab for stability degradation studies, but here mainly applied for CMC. So that means that we're then looking at formulation studies. Besides PK, we do a lot of work that support the development of new or safer contrast agents. So let's have a look at some examples. This is the analysis of transferrin-bound iron. We have done a lot of work for iron formulations. The iron deficiency for different applications. One is iron deficiency, but iron, the same formulations are also sometimes used as a contrast agents, because the formulation exists from very small iron balls, solid iron balls, which are only a few angstrom of diameter. So these iron balls are coming into the hair vessels of the organs, and thereby you can have a very sharp image for CT or MRI scans. There are many iron supplements, and the iron uptake is very complex. So that means that the monitoring of different iron sources is very important. We can also use different proteins, elements bound to different proteins. And for instance, we can use standard kits which have different masses, which you can see here. Here you can see that the peaks are separated for different masses, and you can see that they are behaving linear for a chromatographic application. So that means they have different retention times, So it means that we can, if we have an unknown peak in our chromatogram, by analyzing the retention time, we can then see which of the proteins is present that was used in our application and that binds our element. At the right side, we see an example where we have developed an assay for iron. In this case, we are able to measure the transferrin bound iron, which gives a very sharp peak. And we can now study this peak at different times after dosing. When the iron is given to the patient, we can then see how fast goes the uptake and which part of the iron goes to transferrin or to the albumin bound iron, or which part results in an increase of free iron. And all these different iron sources are now separated from each other and can be analyzed in one run. This is a very sophisticated application. Only a few years ago, this was still done by UV kits, which are based on a coloration of the transferrin bound iron, and these kits were quite inaccurate. So this is a very big improvement for this kind of application. Another imaging enhancement example is gadolinium. Beside the iron already mentioned, we have also iodine, and in this case I have given an example from gadolinium, which is very much used for the enhancement of MRI contrast agents. ICP-MS can then be used to check for the clearance of the free gadolinium. Free gadolinium is very toxic, so we need to have stable formulations. In the past, even there were people killed by using unstable formulations, and the FDA has therefore put very much strict rules on these kind of gadolinium formulations. And even now there is a lot of work in developing more stable, more safe and stable formulations. We have developed an assay here where we can measure the gelated gadolinium. So that is the gadolinium in its safe form, because then it's bound. But we can see at the same time, in the same run, we can see a peak of our free gadolinium. If we zoom in, we can see a very small peak. We can measure the gadolinium to bound gadolinium ratios up to 20,000 to 1 within one analytical run. Besides regular bioanalysis and the examples that I just have given, there are many other applications where we can use ICP-MS. And in the next couple of sheets, I give some other applications. For instance, trace elemental analysis. From the beginning of 2018, the FDA has presented a new guideline where the manufacturers of drugs are, for them it's now mandatory to show that there are no leakage from the container material into the drug formulations. That means these studies are usually called container disclosure studies. Besides the lesion, we can also check for clearance after dosing or the recording of exogenous levels due to pollution. And ICP-MS is very suitable because of its sensitivity, but also because of the possibility to perform multi-elements. As you can see here, we have usually, for contamination, there are usually different sources, and these can then be measured at the same time. An application that is more or less similar to the previous one is limit testing, similar to trace elemental analysis, but now the target impurities are from a specific known source. And for all the different elements in periodic system, the FDA has presented in their guideline a lower limit, which is given for which the application cannot have elements above that limit. For instance, we have done some different studies in our lab. For instance, an example is iodocetamide that is used in an application where iodocetamide is used in diafiltration steps. The iodocetamide may not be present at concentrations above 3 microgram per liter. So that means that the specific sample from that diafiltration step are then measured to show that we are at the safe side. We have in the past done also some work to analyze samples for the presence of palladium. Palladium is used in different organic routes as a catalyst, but you can imagine that after you have synthesized your drug, then your catalyst should not be present anymore. And ICP-MS is a very easy tool to demonstrate that the catalyst levels are below the requested level. But also other contamination from remainders of additives, stabilizers, binders, fillers, etc. These can also be very easy be demonstrated that they are at low and safe levels. Already mentioned a couple of times, multi-element analysis is a very powerful tool from ICP-MS. Strictly say it is not simultaneously, it is in series if you look at the ICP-MS itself, but if you can measure 20 elements within seconds, well, you can then consider it more or less as simultaneously. Applications are, for instance, heavy metals in all kinds of different sources, and biomarker analysis for PD evaluation. Interesting applications are, for instance, Alzheimer and Parkinson. In this case, we have measured some samples where you can measure at the same time the magnesium, iron, aluminium, zinc, and copper. Several of these elements were shown to go upside in concentration and others were going down. This is very interesting to see these differences as biomarker in these therapeutic area. Same for Wilson disease, we measure iron as a biomarker and already mentioned the iron for iron deficiency. And these can be both together measured with some other elements. Also, the specific ratio between different elements that can serve as a fingerprint. A very interesting application that is often that was used or mentioned often in the past is dry blood spot analysis. Dry blood spot has of course many advantages. The big advantage is of course from a logistic point of view. You can imagine if you can take a sample in an easy way as demonstrated here, but just a simple prick in the finger, then the blood is pipetted on a piece of paper, and then the paper can then be punched and analyzed. I think most of you have heard that this development, there was a lot of rumors about it a couple of years ago, and the progress was hampered by the fact that there is a stability issue for the hemochrite. So there is a lot of research going on, but up to now this has not been solved. And I think it's interesting to realize that for ICP-MS, this stability issue is actually not present. If the whole blood is punched, and then the element is extracted with an extraction liquid and then analyzed by ICP-MS, or even digested completely with the paper inclusive, then there is actually no stability issue anymore, because an element cannot degrade. And if you digest or extract the whole piece of paper, then you are 100% sure that all the elements that is in the tube after extraction or digestion, you will measure that by the ICP-MS. For instance, a typical example would be platinum. There are a lot of studies where platinum needs to be analyzed after oncology applications, for instance, to see to check for clearance. Well, this can be done with such a simple application, and then by analyzing the sample. Not mentioned, but here at the right side you can see another application, and this is a laser ablation. That means that prior to the ICP-MS, you have some kind of experimental setup, here a laser, and with the laser you evaporate the sample. That can be, for instance, this part of a paper, and the evaporated sample is then with the carrier gas transported to the ICP-MS. Already mentioned, because there are still no dry blood studies filed yet by the FDA. The progress is unfortunately slow, but at least we hope that in the next couple of years we can demonstrate by using ICP-MS in combination with dry blood spot that this certainly can be a safe application to start. I've now come to my conclusions. Application of ICP-MS is growing together with the increasing number of metals in medicine. ICP-MS is an excellent detector for LC in bioanalysis. Elemental detection is, of course, complementary to molecular MS/MS or UV detection. It enables simple quantification in all kinds of matrices. Little to no sample cleanup is an important advantage, but also suitable for very small amounts of sample. It is a rapid and efficient technique for PK and PD evaluation, and it has a high potential in quantitative analysis, metabolite profiling, immunoassays, microsampling, and it has a fast multi-element analysis which can be of interest for screening. To summarize, LC-ICP-MS is a rising, powerful, versatile bioanalytical technique in drug development. Small volumes important, small tissue samples can be important. For instance, I already mentioned one mouse can be used for one PK profile, and of course, do not forget that it is always complementary in value to the existing techniques, LC-MS and ligand binding assay. Thank you for your attention.
Thank you, Fred. So now we're going to take some questions. Please use the questions box in the control panel to enter your questions. So the first question, Fred, is, are there elements except oxygen, carbon, hydrogen, and nitrogen that cannot be analyzed by ICP-MS, and why?
Yes, I can, if I move to the periodic system, this is, I think, the most easy way to answer that question. Yeah, if you look at the periodic system here, you can see the sensitivity. The most sensitive metals are the blue ones, and then the yellow ones, etc., and yellow. But you can also see, and it was not mentioned, there are also white samples, and these are usually the ones that cannot measure. And of course, there are reasons. Hydrogen and carbon, nitrogen, oxygen are usually considered as not applicable for ICP-MS because they are too less specific. Because in nearly all elements there is one of these elements, but don't forget that even in the air around us, these elements are usually present. And of course, you use argon as a gas, carrier gas in the ICP-MS, but with the carrier gas also normal air is introduced into the ICP-MS. So that means these elements are always giving some background. And then the precious gases here at the right side, they are very difficult to ionize. They have a high ionization energy and therefore are also not applicable to analyze in ICP-MS. And of course, the ones here are usually not stable enough to analyze. But you can see still most of the ones that are the known elements that are present in our drugs, which I have shown in the table, are the ones that are given here.
Great. Okay. In your experience, is it possible to perform lithium determination in human plasma samples for regulated bioanalysis, such as in clinical studies? If yes, which instrumentation and technique do you think more appropriate for this purpose?
Could you repeat the first part of the sentence?
Yes. Is it possible to perform lithium determination in human plasma samples for a regulated study?
Lithium is possible, yes, but it is a very low mass, so that means that the sensitivity is not very high. So it is possible, but then of course lithium should not be used as an anticoagulant. But if that is the case, we can analyze lithium in plasma, but only at relatively high concentrations. So it is possible, but the sensitivity can be a little bit limited. What I mentioned is that at the low masses, you can see here it should be quite sensitive because it's yellow. But these sensitivities are all in clean formulations. So that means, in clean solutions. So plasma is not, you cannot consider that as a clean solution because there are thousands of compounds present in the plasma. That means that there are always some compounds present that can give some interference. And that will usually limit our sensitivity. And the lower the mass, therefore, the lower the sensitivity. So it depends a little bit on the application and the concentration, but it's possible. Okay,
but if you could do it, which technique would you recommend? Again, would that depend upon the application and the goal of the study?
If it is a salt, I think ICP-MS is the only method, although it is also possible to use, for instance, ICP-AAS or AES, so the adsorption or emission machines. But these are, since the introduction of ICP-MS, they are not used much anymore. You can still buy them and they are still used, mostly at the environmental laboratories. But I think LC-MS is certainly not possible unless the lithium is part of a large organic molecule. But if it is a salt, ICP-MS would be the best choice.
Got it. You mentioned using a bio-inert UPLC system. What LC brand is that?
The brand is Agilent.
Okay, great, thank you. And then we have another question here. I guess QPS can use metal salt solutions as standards, and a sponsor would not need to provide analyte standards for PK assay development and validation. Is that correct?
That is indeed correct. We use certified reference materials commercially available. They have a certificate with a certified amount of element, and so that means these are usually used for preparation of the standards. And for the QC samples, we usually prefer the compound from the sponsor, because you know that the QCs should, by using guidelines, always of the same material as that is applied in the drug.
Got it. Should matrix effect be evaluated during method validation?
Sorry, could you repeat it?
Should matrix effect be evaluated during method validation?
Yes, absolutely. Yeah, if you compare between LC-MS and LC-ICP-MS or ICP-MS, all the items that are used, that are mentioned in the FDA guideline or the EMA guideline, these validation items are also applied by us for ICP-MS and LC-ICP-MS. The only thing that is usually not done is the recovery, because we don't use an extraction. But all the other topics are used. And you could even wonder why we should do stability, as I have mentioned that an element cannot degrade, and that is certainly true. But there could be sometimes reasons, for instance, specific adsorption to container walls or to tubings, precipitation, deposition, these kind of things that would also result in a negative bias. The same as for stability, although it is not related to stability, but it's still a situation that you do not want. So you have to prove in your assay that adsorption is not present, or precipitation, or deposition, etc. And the same for matrix.
So essentially, long-term stabilities are also validated, correct?
Yes, that is correct.
Okay. Are there any other questions from the audience? Okay. Fred, if you mind, could you go back to your last slide? Great. I just wanted to thank everybody for your time and your attention. This webinar, like I said, will be recorded and will be available on our website. That's the web address where you can find this webinar in a couple of days, as well as the previous webinars that we have recorded and are available online. And with that, I wish everybody a good day and a safe day. Thank you so much. Thank you, Fred.
Yes, thank you all for listening. Goodbye.
Bye-bye.